Welcome to the first of a three-part lecture. So in this three-part series, we're going to finish our discussions on the modification of Mendelian inheritance, in other words, the Chapter 4 material of your text for genetics, and which addresses non-traditional Mendelian patterns in gene transmission. In this first part, we're going to talk about gene interactions that generate novel phenotypes. I'll give you one example, and X-linked genes. Right, so some genes can interact to produce new or previously unseen phenotypes. This is something that was first documented by scientists Bateson and Punnett in the previous century. They performed a series of classical experiments that demonstrated genetic interactions resulting in new phenotypes. And they analyzed, in order to do this, they analyzed 4 comb shapes typically seen in chickens. And those comb shapes are depicted on this slide. They're single, walnut, pea, and rose. Based on their studies, they determined that comb shape was controlled primarily by 2 genes, which we will call R and P. And I will let you know that still today the molecular basis of those 2 genes is unknown, even though their transmission patterns are very well understood and still used by breeders of chickens today. So in this example, it might be easy to mix this topic up with the topic of epistasis, which we discussed previously. And really there actually is quite a bit of interaction or overlap here. In the case where we just talk about novel phenotypes due to gene interactions, and we leave it at that, and we don't talk about the underlying molecular basis, it's typically because we don't know what the underlying molecular basis is. And if we don't know the underlying molecular basis, it becomes very difficult to actually describe epistasis properly. So let's think a little bit about these chicken inheritance patterns. What Bateson and Punnett showed is that when you take 2 true breeding parents that have the pea or the rose comb shape, and those parents are shown up here in this row. This is the parental generation, okay? And they're true breeding, so they're homozygous for each of the 2 genes. Big P, little r, and so on, okay? So the gametes that these parental organisms can show or can produce are shown here, okay? And then when those gametes fuse to form the F1 generation, what they discovered was a walnut-shaped comb, and I showed you a picture of that on the previous slide. So in the heterozygote for each of these genes, you get a walnut comb. So it's a new or a novel phenotype. And they were really intrigued by this, okay? What they did next is they got an even more interesting observation in that they let those walnut-combed F1s go ahead and reproduce. So they looked at the F2 generation, right? And these walnut-combed individuals can produce the gametes that are shown here and here, okay? And you know how to describe those. And then this 4x4 Punnett square is showing the possible genotypes and the observed phenotypes that these offspring produced. And what they discovered is that in the F2, they got an additional phenotype, and that additional phenotype was the single phenotype shown here, okay? And there's a picture of that comb shape on the previous slide. So they did see a 9:3 to 3:1 ratio in these offspring, and that should remind you of the typical patterns seen in dihybrid crosses that Mendel observed and documented so well. But what's different here is that the 4 phenotype categories include 2 that are novel, and those novel phenotype classes are the single and the walnut. So single and walnut were not present in the original parental generation, so they're considered novel phenotypes in this study. And while Mendelian inheritance explains the genotypes, it doesn't really explain the underlying phenotypes properly. So this is a very classic example of where we know that gene interactions can result in novel phenotypes, but as I said a moment ago, we don't know the molecular basis underlying this particular genetic story. Another example or genetic story where we see inheritance that doesn't follow the typical Mendelian patterns that we've discussed earlier is the example of sex linkage. So in order to talk about that, we're going to need to talk about the discovery of sex linkage, which is something that happened primarily in Thomas Hunt Morgan's lab around 1920. And in that lab, he had a suite of geneticists working and in training with him, and they all focused on the model organism Drosophila melanogaster, or the fruit fly, which is shown on the left. Now, to discover sex linkage, that was discovered through studies that involved a particular mutant, and the mutant is shown here. And this Drosophila is different from the wild type, which is shown here, in that it has white eyes. It has no pigment in the eyes. A typical wild type Drosophila will have this kind of reddish brick coloring in its eye pattern. So let's think about some of these experiments, what they meant. So Morgan and his colleagues were really interested in the white-eyed flies, and what they noticed is that the white-eyed flies would just show up every so often in their Drosophila collections or cultures. And so they set out to try to better understand the transmission of this white-eyed phenotype. And in this first experiment that was conducted in Morgan's lab, what they did is they found red-eyed females, so there's the female symbol here, okay, and they crossed them with white-eyed males, okay, and that's the phenotype shown there. So at the genotypic level, we know now that females in Drosophila are XX and that males are XY. Okay, we'll talk a little bit in future lectures about the sex chromosomes in Drosophila melanogaster. The W gene, which is given the symbol W, is shown here and here. Okay, and the little plus symbol implies that it is wild type. Okay, over here on the right, this male fly is an XY. The Y chromosome lacks the W gene. The X chromosome that this male has the W gene, but it's the mutant white-associated allele. Okay, so that's the genotype, and the corresponding phenotypes are shown up here. So when those organisms go ahead and they produce gametes, you will get the gametes that are shown in this part of the figure. So the female is going to be able to produce two identical gametes with regards to the X chromosome, shown here and here. And both of those X chromosomes are carrying the wild type W gene associated with red eyes. The male, in contrast, is going to be able to produce a gamete that carries an X chromosome, and the X chromosome in that particular male, or from that particular male, will have the mutant W allele of the white gene that is associated with the white-eyed phenotype. And of course, he's also going to be able to produce gametes that carry the Y chromosome. When those gametes fuse, the possible F1 progeny are shown down here. So you can get these females and these males. Okay, so the F1 genotypes and phenotypes are a half this and a half that. Okay, here we've got the females and here we've got the males. Okay, and the phenotypes are half female, half male, and all of them are red-eyed. And so the fact that they were all red-eyed is something that, in Morgan's lab they would look at and they would think, oh, well, the F1 are uniform with regard to eye pigment. So that's the principle of uniformity in the F1. That's something that's very common in Mendelian genetics. But let's think about what happened next. Like all good Mendelian geneticists in Morgan's lab, they wouldn't stop with the F1, but they would then continue to go on and look at the F2. Something you can do very quickly and easily in fruit flies because they reproduce, in periods of time that are roughly 2 weeks. So here is a continuation from the previous slide. These are the F1 organisms we just looked at, okay? These are their phenotypes, and these are their genotypes, okay? So all of the females are going to be heterozygous for the W gene. The males only have one X chromosome. That's a situation referred to as being hemizygous, H-E-M-I zygous. There's only one copy. Okay, it's a term we use to apply to sex chromosome situations. So the male and the female are red-eyed, but their genotypes are different. the male is red-eyed in that his only X chromosome carries the wild type red-associated W gene, and then he's got the Y, okay? The female is heterozygous. She has two X chromosomes, and on those two X chromosomes, she has one of them has the wild type W gene, the other has the white. Okay, now what happens in the F2? So when these gametes, which are the gametes that can come out of the F1 organisms, okay, assuming that you're crossing the red-eyed female with the red-eyed male, okay? Right? So we're carrying on. Now let's look at the F2. So these the gametes from the F1 are shown here and here, and this is the F2 generation. And look what's happening. We've got females on the left and males on the right in this Punnett square, this 2x2 Punnett square. The females here are red-eyed, and they are homozygous for the wild type allele of the white gene. The females here are red-eyed, and they are homozygous for the, heterozygous for the two different alleles of the white gene. Here we've got red-eyed males and white-eyed males. The red-eyed males are red because their only X chromosome contains the wild type copy of the W gene. The white-eyed males are white-eyed because their only X chromosome contains the mutant white-associated allele for the W gene. So we're seeing something a little odd here in that we can say that we have a 2:1:1 phenotype ratio. 2 parts red females, 1 part red males, and 1 part white males. That's not a pattern that is typically associated with Mendelian patterns. So this is where things are, a little bit different. Also, like all good Mendelian type geneticists, in Morgan's lab, when they were doing these experiments, they would be concurrently doing the reciprocal cross. And so the reciprocal cross in this story, it's shown here. And here we're going back to the parental generation, and we're flipping the phenotype of interest and the sex that it's found in. So in this cross, in this scenario, the reciprocal cross is where you take a white-eyed female and you cross it with a red-eyed male. The example or the story that we just looked at, the experiment that we just looked at, you had a red-eyed female crossed with a white-eyed male. Now we're going to do the opposite, okay? And so the two figures that are shown here on the left and the right-hand side show the outcome of that reciprocal cross. And you can take the time to walk through this. I think it's pretty self-explanatory. But the point here, the punchline, is that the F2 from this reciprocal cross are showing us a different phenotype ratio than the F2 in the previous example. Okay, so the F2 phenotypic ratios are different between reciprocal crosses. And that's something that is commonly associated with sex linkage. So in this example, we've got a 1:1:1:1 ratio, red-eyed female, red-eyed male, white-eyed female, white-eyed male. And you can see why that would be the case by looking at the genetics here. So what Morgan's lab published out of all of this work is they published this study and they documented it as an example and a description of what is now known as sex-linked inheritance, which is the inheritance of a trait due to a gene linked to a sex chromosome. That example that we just looked at was the inheritance of the trait of eye color, and it was associated with the W gene, which is associated with the X chromosome, a sex chromosome in Drosophila melanogaster. Sex-linked inheritance is usually discovered through the use of reciprocal crosses, and it's also identified by looking for patterns and pedigrees where traits seem to track with increased frequency with one sex or the other. Because sex linkage alters traditional Mendelian patterns of inheritance, it's included in this Chapter 4 discussion. Now I'd like you to go on to the next part of this lecture.